All right, let's give a warm applause and welcome to Dr. Kasri.
So thanks a lot for sticking around, and it's really, for me, a pleasure to be here. So it's the first time that I'm attending a summit, but I'm actually already working, I think now 5 or 6 years almost, with Dr. Kohn and Bert de Vries on the research side of the disorder. And I would like to just share some of our findings with you and especially show you how we approach the problem. And it is quite different because we don't really work directly with the patient. So for me, it's really been an amazing privilege and really special to the past 2 days to also meet you, the parents, but also the kids, because otherwise I don't have this opportunity. So since I arrived, several people asked me what I'm doing. So how should I call you? So I'm a doctor, but I'm not a medical doctor. I'm a neurobiologist. And a neurobiologist is actually interested in how the brain works. So I'm interested in the biology of the brain. So we try to look at the brain, we try to model the brain, we try to build up the brain from scratch, and we try to take it apart to see how it's working. And so a few years ago, or I don't know, David, how long time ago it was, but David approached me and he said, you know, we have this this disorder that we just identified, and we actually don't understand why a mutation in this gene, cancel-1, actually causes the disorder. Would you be interested in tackling this problem? And so I agreed, and so I learned about Kohn-de Vries syndrome, and I learned it's a multisystemic disorder, but where the brain is among one of the targeted organs. So what we did not know after the identification of the gene is what the gene actually does in the brain. And so we actually, in my group, have 3 very well-defined questions. Is really try to understand what this gene is doing. We want to understand, so that's in a normal situation, so what is the function of this gene? We want to understand why it is when you have a certain mutation that you have the syndrome. And the third one, which is probably for you the most interesting one, is can we rescue? Can we do something about it? Can we rescue the phenotypes we see? So those 3 questions are actually 3 very hard questions, very difficult, especially if you think how the brain is formed. So we have in our brain about 86 billion nerve cells. And I think you can arguably say it's the most complex organ that we have. And so here you see that the brain consists of those nerve cells. And those nerve cells, they are very particular because they communicate with each other by sending electric signals. So they do that all the time. And that's actually the way our brain works. So it's not one nerve cell, but it's those 86 million cells that continuously communicate with each other. And every time they do so, they basically have this electric pulse that is used to achieve this communication. Now, you can imagine that that's quite difficult to study. So what we have to do in the lab is really ask the question, how can we now do this in the lab? Because I can't go into Bert's brain and try to figure out what makes Bert. And I can't do that with your kids, right? That's very clear. We can't do experiment with the human brain. So we have to find a way to do this. And luckily, we actually can do this. And it's not so long time ago. So since 2006, we now have the means to actually build a human brain, or at least a very simplified version of it, in a lab dish. And I want to take you to some of the terms and the things that you have already heard, just to explain how this works. So what we basically have to do is we have to start with a stem cell. And the stem cell is actually you could say, the origin of life. So this is a cell, and everybody starts by being just one stem cell. And a stem cell has a property that it can divide multiple times, so over and over, continuously. So you can make a lot of stem cells. Now, this is really required because ultimately we want to form a whole person. Now, the second property of a stem cell is that it can basically become any other cell in your body. So stem cell can become, for example, a nerve cell or a muscle cell or blood cell. So that's actually the start of what we can use now to try to generate those nerve cells. And the way it works is actually best illustrated with this analogy. We are in Park City, so it's a lovely region to ski. So just imagine that you're on top of the mountain, and on top of the mountain you are like the stem cell. Now when you're on top of the mountain, you often have different opportunities to choose which valley you want to ski in, right? So a stem cell, basically, when it gets a certain push, it can choose to go in a certain valley. Now once it goes down that valley and comes all down at the bottom, it will basically change into a nerve cell. Yeah. Now, the problem is that in all adults or grown-up people, we don't have access or very difficult access to those stem cells. So what we actually would like to do is to generate those nerve cells, not coming from the stem cell, but from other accessible cells. And we have been privileged that yesterday some of you have donated some cells. They have donated some skin cells, I believe, right? And so the question is, what are we going to do with those skin cells? So the trick is that we want to take those skin cells and we want to make them nerve cells. And the way we do this is, it's not an easy thing because, again, if you take the analogy of the valley and skiing, if you're in one valley and you want to go to the other valley, no, that's not easy. So the only way you can do this is actually to go way back on top. But you can't do this, right? You can't ski back on top. So about 10 years ago, 10 years ago, some very clever scientists, they actually figured out a way to go back uphill. And just see it, they basically invented the chairlift, right? That's what they did. So here we have now, basically from any cell type that we can start, we can take the chairlift, we can go back up the hill, and from there on, because it's a stem cell, we can again choose which cell we make. And in our case, we are particularly interested in making those nerve cells, because those are the ones we would like to study. So this is kind of a little bit the idea of how we, why we collect your skin cells and what we do with them. Now ultimately we want to then do this from the patients and healthy siblings or parents, because we can convert them to stem cells, to neurons, which we then can compare with each other. So, and these are actually how the stem cells, or how the nerve cells look like in a dish. So, Bert was saying about his favorite movie, was saying this is one of my favorite figures that I like to look at, because here you see this typically from morphology or shape of a human neuronal cell. So this is a cell that we have grown in a dish that we now can study. We can look at the morphology, but we can also look at many of them. And here you see, I don't know how many there are here, but this is actually a whole collection, thousands of those nerve cells. And the interesting thing is that, like I showed you in this little movie, also these nerve cells, they communicate with each other. They send electric signals to each other. And that's, of course, very powerful, because now we can use those signals to really look at how neuronal cells, nerve cells, communicate with each other. So the general strategy that we take then is that we can compare nerve cells from healthy controls versus the nerve cells from your kids. And of course, the biggest advantage is that every of these nerve cells, even we grow them in a dish, in a petri dish, in a lab, has actually the exactly same DNA of genetic information as the donor, so as the parent or the child in this case. So this gives us a very nice model. And this is fundamentally different than what we will do, for example, with a mouse model or a fly model, where we have to make a model, but by making this model, we take actually a lot of away of the really essential information that is coded in the DNA of the patient. So now this is how we generate nerve cells. The second question is really, what do we do with it? What do we measure? And I already alluded to it. We like to look at the communication between the neuronal cells, because the hypothesis is, and we have shown that with other disorders, that actually the communication probably between the nerve cells is impaired or is different. And we'd like to know how this is. So the way we do this, and Bert really scooped me on this already, but I think we can just repeat it, is that we can grow those nerve cells in what I like to call smart dishes. And those dishes, they are smart because, as you can see here on the bottom, you have those little black dots, and those black dots, they are actually just electrodes. And those electrodes, they can sense the electric activity. So this is, you have already seen before, we call this a typical a chip, and that's why we call it brain on a chip, because we can now really grow the nerve cells on this chip. And what's great about this chip is that basically it will measure the spontaneous activity that is going on. And you can do this in a lab, you can grow them, the nerve cells, and you can measure that every day. And every day you will see that the communication will increase and will change. And so in a way we can mimic the development of the nerve cells of every individual. So in principle we can do that from everyone here in the room. I mean, it would cost quite some money and some time, but we can do that. And that's a movie that Bert likes. And so I want to give some explanation about the movie. So every white dot that you see here is an electrode. And whenever the color becomes red, that means that nerve cell that is sitting just on the electrode is what we call firing in action potential. So it's gives this electric pulse. So you see, I think you can see that very often the whole image becomes red, right? What does it mean? This is actually translated in these vertical black lines. You don't have to worry too much about them. But what cells like to do, nerve cells like to do when they develop, is that initially they are like active on their own. They are just like, I'm firing an action potential, and then the other cell is also firing action potential. But as they grow older and they start to mature, they like to be active together. Just like we as humans, we also like to be in a network. We all like to be on Facebook and other networks. Those cells also like to be in this kind of network. And this is really, for us, a measure of a healthy or normal development of these cells. And so if you really want to translate that, by looking at this kind of activity, we can see when nerve cells are communicating, how often they are communicating, with who they are talking, and also how loud they are talking. So this is an extremely powerful platform now to do this and compare the nerve cells from the healthy individuals versus the KDVS kids. So how does it look like then if you look at this kind of pattern from a healthy individual? So here you actually have show three of those plots. And the only thing you need to take from this is that those three plots, they look very similar. Right? So you see that these vertical lines, they are extremely regular, and you have about the same amount in each of those healthy patients. So think about, so those are three completely non-related individuals for which we took fibroblasts or skin cells, like we did for some of you yesterday. Then they went through the whole process of making those stem cells. Then we generated those nerve cells, and then we measured them. And at the end, we have this very, very similar mode of communication between those healthy patients. So the question now is, how does it look like for colon de Vries syndrome patients? Is it also looking like this, or is it looking differently? And so this was work that has been done by Catherine, and some of you remember Catherine. She was here 2 years ago in Memphis, and she will be here. She arrives tonight, and she will be here tomorrow giving some more details about the work. But what Catherine has done is she basically compared the profile, the electric profile of the control versus KDVS kids. And so I won't go into too much in detail, but I think that you can already visually see really the difference between the control and the KDVS kids, in the sense that we see less frequent communication between the nerve cells. We see that this communication is less regular, and it's also shorter. So there is, in general, less activity in those neurons. Now, this was actually a very important finding for us, because it, for the first time, gave us really a neurobiological readout of what's going on in the brain, actually, of what we think is going on in the brain of Cohen de Vries patients. And this, of course, we have done that for Cohen de Vries patients, but we have also done this for other syndromes. And what is very striking is that for each of the syndromes, we have a signature. So those properties here is actually something that we see very typical for the Cohen de Vries syndrome patients. Now, yeah, this is something that we can see and we can observe. What can we do with it? Well, we think that this is a very important first step to try to understand the disorder, but also try to find approaches or rationales on how to basically correct or improve the communication of the neuronal cells. Because what we ultimately want to do is to go back from this situation to a normal situation. And Catherine will show you some really interesting results that allow us to do this. And of course, that's also the strength why we have those nerve cells in a dish, because we can try out a lot of things. We can try out certain compounds, all kind of manipulations, which we of course cannot do on a human being. And that is actually what we are planning to do. And that's also why we got more skin cells, so we can make more, test more of those, or generate more of those profiles for more kids. But ultimately, we want to understand what is really causing this lower activity in the nerve cells, and can we do something about it? And so this is a scheme here, I think, which is always, it just gives you a sense of the different steps that we need to do in normal basic research to come ultimately to, hopefully at some point, something that is also really useful for you. And at this moment, we are really doing the basic research, where we try to identify certain phenotype, but also certain targets, compounds that can ultimately change the profiles. And so we are doing, at this moment, compound screening. So because we have those cells in a dish, we can add a lot of compounds. And this is based on quite some rationale that, again, Catherine will tell you about tomorrow. And this we can all do in a dish. And our expectation is that we will find some compounds that can revert the phenotypes. But ultimately, of course, this is something we see in a dish. And ultimately, we want something that really can help patients and also works on a full brain. So not on a simplified version. And for that, there will be what we call the preclinical validation of any compound that we can find. And that's something that we ultimately need to do on a complete brain. And for that, we use models like the mouse model, which Bert already alluded to, and that shows, for example, this high sociability. So, and that's something that we hope to be able to do in the next phase. So we are now currently doing this compound screening. We have some interesting results that Catherine will talk about. But then the next step will really go to the behavior of, to test whether these compounds will also work in a mouse model. And also for that we are preparing, actually, because we need to be prepared in testing the compounds. And well, we know that now the kids have certain behavior which we can really also translate into the mouse model. So those are social interaction, vocalization, and also learning and memory behavior. And that's something that we can now actually do in a lab. And that's something that we've been building together with Bert in some tests that allow us to track the mice. And what you see here is actually all kind of behavior that the mice can do. And mice like to be together. And what Bert showed you before was really a test where you see that the mice would interact with another mouse or not. But those are kind of enforced behavior. And it's always you test one mouse. What we want to do now is actually look at how those mice live together and how they have their social interactions in a more spontaneous, normal habitat. And why we do that is because we can see that mice actually also exhibit quite complex social behavior. And so you would like to track this in a kind of natural habitat. And for that we have been building this live mouse tracker. And this is how it looks like. Is a big cage with here an Xbox camera. So you see we like to use all this technology from gaming. And the advantage is that the Xbox camera can actually track an individual mouse. And it actually looks like this. So this is a top view of this live mouse tracker. And you can identify 4 different mice here. Black mice, 1, 2, 3, 4. And I will just play a movie, and what you will see is that the camera will start detecting the mouse. And by doing so, it will start also really trace or track the mouse as it moves around the arena. This is the arena. And by analyzing the 3D movement of the mouse, it will basically be able to tell us whether a mouse is interacting with each other, whether they are making a group or breaking out a group, or whether they go nose to nose or nose to tail, and so on and so on. So I'll just show you. I hope that the movie works. So here you see the different mice. So you see that he has detected one mouse here, a second mouse. So the 4 mice are detected. And then you can see that we now can track those mice all the way through the arena. And we actually don't have to do anything. Those mice, they are like in their cage, and we just film it. And then automatically, all the behaviors that I showed you on the previous slide, all those complex interactions, they basically roll out of the computer. And this is what we actually want to use as a phenotyper, called this phenotyper, to first also see what's different between the control mice and the mice that lack the cancel 1 gene, but ultimately also to see whether we can rescue certain type of behavior. So I hope that with this I kind of gave you a little bit of an idea of what we are doing in the lab, and hope to see you next time in 2 years. Not sure where it will be, but
So we have some time for just a couple questions before we head out for lunch, and I see one right over here and one back there, so.
A question, my son has seizures, and seizure meds, of course, also affect how the brain works.
Yeah.
Is this kind of neuronal activity something that you created this, test brain cell, something you can also test how the seizure meds are working on neuronal activity as well for KDVS individuals?
Yeah, so the question is whether we can test the influence of epilepsy meds on the normal development or how it affects the KDVS neurons. That's exactly what we can do. So that's exactly what the big advantage of having those cells in a dish is that we can try to look at what the effect is of the regular drugs that maybe some kids get, but also we can see if kids that might have epilepsy, because not all the kids have them, whether we can see this also in this test, and whether we can, of course, then remediate that with certain typical anti-epilepticas that are being given. So that's exactly the advantage and the reason why we do that. Yeah.
So I don't know if she's gonna talk about it tomorrow or not, but my question was, and hopefully I can say it, what my mind is thinking. So the stem cell result that you had of the control versus the KDBS, that it was slower, shorter, and irregular.
Yeah.
On that slide, what does that actually translate into what does that actually come out and mean in the behavior or the
Yeah.
function of the child? Is that,
Yeah.
hopefully I get my question.
So the question is, how do we translate now what we measure in a dish to behavior? Right? So that is a very big step. Right? So what one needs to be aware is actually what we want to create is something what we call an endophenotype. We want to have something that a phenotype that we can measure and we can work on, but we cannot translate directly the electric signal to behavior, because behavior is so complex and requires many different areas of your brain to talk to each other. So the only thing that we can deduct from this is that there is something in the individual cells that is different, that makes that they will communicate less, so that the action potentials are not going as they should. And so it's a simplified system, but it's actually a great system because it allows us to try to correct for it. But what one should not do is really try to translate that to behavior. So you should not think that, they are less active, that the kids are less active, and so there is not this one-to-one. It's purely the inherent property of the cell that we are looking at in a way. Yeah.
Get my steps into there.
So I'm curious about the role of cancel 1 in all of this. So is there evidence that cancel 1 is more active is used in the brain more than other body systems? Or do you have any idea of a link between the loss of function of cancel 1 and what you're seeing in your results?
Yeah, so so the question is, what is cancel 1? How does it really contribute to this phenotype that we see? So that's actually the work that Catherine has done really in detail, and she will talk about it. We find that in the brain, cancel 1 is very important in this kind of process that what we call autophagy, which is a very complex word, but basically your nerve cells need to recycle all the material during its whole life. Because nerve cells, they have the properties, they don't divide. So once they are born, basically they have to last their whole life, they have to last, right? And so they have to recycle all the time material that is required to keep the cell alive and well functioning. And we found that cancel 1 is actually important for that process. So if you miss cancel 1, the whole recycling system is not going as it should be. And that will ultimately lead the cells to be less active. And that's something that is really, it's important to know, because then we know what we need to target to try to make this recycling going up again. And that is actually, I think we'll talk a bit more in detail about that tomorrow, but that's the short version of the answer. Yeah. Yeah.
I'm just curious. It seemed like the slides were all referring to the gene mutation and not the microdeletion. So are you only focusing on the mutation?
No. Good question. So is this only for the gene mutation or for the deletion? So luckily we also received some skin cells from kids with the deletion. And what we find is that we find exactly the same phenotype whether you have now the mutation or the deletion. So we don't find a difference between that. So we can treat them the same way, and we find the same phenotype and the same recycling pathways affected. So that's almost really one-to-one. Yeah.
All right. Thank you.